Blood is a specialized type of connective tissue in which the fluid intercellular substance is called plasma. If you remember from the previous lesson, classification of tissues, blood is one of type of the connective tissue. As the first, it's a tissue of the internal environment. And let me remind you structure of each type tissue. It's composed by main structures, main units, like cells and the postcellular structures and acellular structures. So blood and intracellular matrix. So blood is composed by cells, forming element of the blood, and the intercellular matrix, which isn't composed by the ground substance and fibers. It's composed by only ground substance, which is named plasma. Quantity of blood of an adult man is 5 till 6 liters. Of adult women 4 till 5 liters. It's about 7% of the body weight. What about functions of the blood? The first function is the transportation of gases, oxygen, carbon dioxide, different nutrients. So generally it's a transportation of different substances which is necessary for living of organism. Next function, regulation of the pH. Next one, third one, restriction of fluid, losses and injury sites. Next, defense against toxic and pathogens. Blood transport, white blood cells, specialized cells which take an active part in immunity. And this can make transportation of different necessary substances like antibiotic. It can make transportation of antibodies, special proteins, which explain humoral immunity. And last function, it's not last function, really, it's a next function, stabilization of body temperature. So we explained main functions. Blood has a lot of extra functions, but we don't have enough time to explain all of them. Let's go to component of the blood. Component of the blood are explained by the formant elements, like cellular elements, which constitute about 40-45 of the total volume of the blood. You can see here I have 40 till 45. An average in men 40, 48, in women 36 or 42%. It's a percentage of formant element of the blood to the plasma, or percentage of the plasma. So this percentage of formant element of the blood is named hematocrit. And formant element of the blood is such as erythrocytes, leukocytes, and platelets. We couldn't call them cells. Red blood cells and white blood cells, it's an incorrect name because, for example, erythrocytes and platelets with another named cells, really they are not cells. They are postcellular structures. We couldn't call them cells because, for example, erythrocytes don't contain nucleus and organelles, and platelets, there are pieces of cytoplasm and cell membrane, so it's like incomplete cells, and the pieces of cells. And so that only leukocytes are cells, so that we couldn't call of them cells of the blood. We can call them formant element of the blood. Let's go to the structure, which come back to the structure of the plasma. Let me start from the structure of the plasma. About component of the plasma. Component of the plasma are water. We take about 92%. Organic components, we take about 9%, and 1% inorganic salts. The main component of organic substance are proteins. There are primary classes of plasma proteins are albumins, globulins, and fibrinogens. Three main type proteins which create 9% of the plasma. Albumins constitute about 60% from them. Albumins are main component, and the albumins make osmotic pressure of the blood. It's a very important function for the human body. Albumins are also very important because the ability for transportation. They are transport proteins, and they can bring different necessary substances such as fatty acid, thyroid hormones, some steroid hormones, and other important substances. Next type proteins, globulins. Take about 65% of the total volume of proteins of the plasma. Examples of an important plasma globulins such as antibodies and transport globulins. Antibodies, also called immunoglobulins, they are produced by plasma cells, and they take an active part in the humoral immunity. And the transport globulins, they also very helpful for transportation different necessary substances. And last type proteins, fibrinogen, and functioning clotting. And the fibrinogen, it's a passive kind of the protein. It's soluble and is the influence of activation substances which are released by platelets and the endothelium of blood vessels. They became fibrin. Fibrin, it's an active kind, it's insoluble. So blood clots are composed by fibrin, platelets, and aggregated formed element of the blood. They take an active part in prevention bleeding during the mating of walls of blood vessels. Element of the blood. As a first, it's erythrocyte. Let's start from erythrocytes. Erythrocytes or red blood cells. The no name red blood cells, but it's not correct. It's less correct than the erythrocyte, because real erythrocytes are post-cellular structure. During differentiation, they lose nucleus and organelles, and they became post-cellular structure, because if you remember, we couldn't call structures which don't contain nucleus and organelles cell. We can call them post-cellular structures because in the first part of the lifetime they live as cells, and after which some accident would happen in their life, they became post-cellular structures. And the erythrocyte are post-cellular structures which have no nuclei. These cells give the main function of the red blood cells or erythrocytes is the transportation of gases, oxygen, carbon dioxide transport. Erythrocyte consists only of the outer plasma membrane, and they enclose the special iron-containing protein hemoglobin. So during maturation, during differentiation, erythrocyte produce hemoglobin by the rough endoplasmic reticulum, and during lifetime, because of lack of space inside, erythrocyte push from the cytoplasm nucleus as the first nucleus, and the next all organelles together with rough endoplasmic reticulum. And finally, finally they contain inside only granules of hemoglobin. So that they are not cells, they are post-cellular structures which mainly composed by hemoglobin granules and cell membrane. What about count of hemoglobin? The normal concentration of erythrocyte in blood, you can see here count of hemoglobin. In men, since 4 till 5 multiply 10 upgrade 12 per 1 liter. In women, 3.9 till 4.7 multiply 10 upgrade 12 per 1 liter. Women have less count of erythrocytes than men because female sexual hormones can inhibit gamepoiesis. Erythropoiesis is a process of formation of erythrocyte, and because women every month lose a little bit of the blood, so because of menstruation cycle, and so that women, it's usually normal, they have a less amount of erythrocytes than men. If we have in the peripheral blood more than average level of erythrocyte, this condition is named erythrocytosis. If we have in the peripheral blood less than average count of erythrocyte, this condition is named erythropenia. Erythrocytosis and erythropenia, they are not normal condition for the human body. And let's go to the next characteristic of the erythrocyte, I mean size and shape. As you see here, the average size of the erythrocyte about 7.5 nanometers, and if you have the average size, if you have size of erythrocyte more than 8, we call such type erythrocyte like macrocyte. If erythrocytes have less than average size, there are microcytes. And about shape of erythrocytes has shape the biconcave discoidal shape, biconcave. Why biconcave? Biconcave, from both sides erythrocyte has evagination, because it doesn't contain nucleus, so that it has this evagination. It's very helpful for transportation of erythrocytes in very, very small capillaries. But possible present in the peripheral blood erythrocyte of different shape. Look here in the peripheral blood, we can have till 25% of different shape erythrocyte. If we have more than 25% of erythrocyte in the peripheral blood, such type condition, it's a pathologic condition, it's named poikilocytosis. What about size? If we can have in the peripheral blood in normal till 25% of different size erythrocytes, macrocyte and microcyte, till 25%. If we have more than 25% of different size erythrocyte in the peripheral blood, this condition also is a pathologic, it's named anisocytosis. Let's go to the characteristic of the hemoglobin. First of all, what about concentration of hemoglobin? Concentration of hemoglobin in men about 135 till 180 gram per 1 liter. In women, about 120 till 140 gram per 1 liter. Condition if we have less than average concentration hemoglobin per 1 liter in men and women is named anemia. It's not good for the human body because anemia brings ischemia. So it means some places of the human body, and mainly the peripheral places, don't get enough volume of enough concentration of the oxygen. They cannot get a normal concentration of different trophic substances. It can bring cell death in these places. Hemoglobin is composed by the hem and globin. Globin is an organic component, it's a protein. Hem it's an inorganic component, it's an iron. Globin is produced by the erythrocyte. During maturation, when erythrocyte contains rough endoplasmic reticulum, it produces and accumulates globin. And iron erythrocyte get in the bone marrow, being in the bone marrow, get from macrophages. Macrophages bring this iron, they get iron from the liver, and not from the spleen and from the intestine. So if we eat food, we get together with food we get iron, alimentary way. And the erythrocyte, the average lifetime of erythrocyte is 120 days. To finish lifetime, they go to the spleen where happen death of erythrocyte, and macrophages digest them, remove iron from old dead erythrocyte, and bring this iron to the bone marrow. Bring to the bone marrow, give new young erythrocyte for formation and production of the hemoglobin. So it's a cycle, circulation of iron in the human body. And hemoglobin depends on the main function of hemoglobin. Hemoglobin hold gases, and because it's make a connection with gases, because of that possible transportation of gases by the erythrocytes. And depending on the quality of gases, we have some different type of hemoglobin. It can be carb hemoglobin, oxyhemoglobin, carboxyhemoglobin, depend on the gases. Yes, so erythrocyte get oxygen in lungs and bring them to peripheral, to different part of the human body, and get CO2 and bring it back to the lungs. Yes, let's go to the next formal element of the blood, which is a leukocyte. Leukocyte white blood cells. They are really cells because they have cell membrane and nucleus and cytoplasm, so we can call them cells. And it's composed by different type of cells. But a count of leukocyte is 6 till 9 multiply 10 upgrade 9 per 1 liter. If we have more than average level of leukocyte, such condition is named leukocytosis. If we have less than average level of leukocyte, this condition is named leukopenia. Leukocytosis is a condition if you have more than average level of erythrocytes. It can be explanation of the exhibition of the inflammation. During different infection diseases, during different type of inflammation, we have increasing of level of leukocyte. What about leukopenia? If you have a less than average level of leukocyte, leukopenia possible during different blood diseases. What about classification? Some type of leukocyte contain the cytoplasm specific granules, and other type don't contain any granules. So, depend on the presence or absence of granules, leukocyte can be classified to granulocyte and agranulocyte. Granulocytes such as neutrophils, eosinophils, basophils. Agranulocyte, there are lymphocyte and monocyte. Let's start from neutrophils. Okay. Neutrophil granulocytes. Neutrophils constitute about 65 till 75%. So total percentage of neutrophils 65 till 75. But they can be classified depend on the type of maturation, depend on the level of maturation. They divided to young, band, and segmented. Young, there are kidney-shaped nuclei and their cells. They contain kidney-shaped nuclei, and they constitute about 0.5 till 1% of the percentage of neutrophils. And next, band. They constitute about 1 till 6%. And band cells have non-segmented stick-shaped nuclei. And the last type, segmented, would constitute 47 till 72%. and have segmented nuclei. A segmented nuclei like this. It's depend on the condition of nuclei, tell about level of maturation. So young, band, and segmented. Young, they are youngest, and segmented, they are mature neutrophils. What about lifetime? Neutrophils are short-lived cells, and they live about lifetime take about 6-7 hours in blood. After they can migrate to different tissues and to the spleen, and where they can live since 1 till 4 days. And what about functions? Functions, so they take an active part in different processes of inflammation. Taking part, they work like, sometimes they work like killers. So they are like macrophagic cells. So they can digest some pathogenic agent we enter to the organism. Neutrophils. And in female, inactive X chromosome appears as a dream stack evagination or appendant of the lobe of nucleus is named bar body. If anywhere you find the short evagination of the nucleus, or like here is it. It's a nucleus and it's a very, very short like point or like stick evagination of the nucleus. It means there is a bar body. So it's exhibition of a female, it's indication of the female blood. Only female blood has this bar body in neutrophils. Eosinophils are named eosinophils because of present a lot of pink color granules, which are colorated by eosine. Acidophilic granules or eosinophilic granules, pale color. They take about 0.5 till 5%. They are small cells, and they are eosinophils. Yes, they are small cells. And what else? Specific eosinophils also. They take an active part in immunity. Especially, they can you can find a lot of eosinophils during allergic reaction. It's increasing of level of eosinophils in the peripheral blood. More than average, it can be exhibition of allergic reaction. Lifetime of eosinophils about 4 till 5 days circulation in the peripheral blood. After that, eosinophils can migrate to the connective tissue or different organs which contain connective tissue and can still here for 8 and 12 days. I talked about different microphagocytosis, and they take an active part in immune reaction, so help to main cells to digest allergic agents. Basophils, eosinophils, basophils. Basophils or basophilic granulocyte take about 0 till 1%. And we call them basophils because of presence of basic staining granules, basophilic granules, basic staining granules, dark blue color granules in the cytoplasm. And what about structure of these granules? The main component of these granules it's heparin and histamine. Heparin is an anticoagulant which prevent blood clotting, and it makes blood more liquid. And histamine is a mediator which can increase draining of the blood through the wall of blood vessels. So it's like increase interstitial fluid. It helps to plasma of the blood leave the blood vessels. So it's increased tumor. And the T connective part basophils take connective part in different inflammation reactions. So if you remember exhibition of inflammation, it's a tumor, color, red color, temperature. So tumor is made up of the interstitial fluid, and the components of granules of basophils very helpful for formation of this tumor. So both components increase draining of the plasma, of the water from the plasma to the interstitial fluid. Basophils. Lifetime of basophils about 3 days in the blood, and then they migrate to the tissue, mainly in the connective tissue, and they became tissues basophil. So the next group of leukocytes, there are agranulocytes. Agranulocytes, there are leukocytes and monocyte. A lymphocyte and monocyte. A lymphocyte take 19 till 37% of all volume count of leukocyte. A lymphocyte can be classified depend on the size, to the small, large, and depend on the type, to T and B. T, it means T-dependent cells, T-dependent or thymic-dependent cells. We get differentiation in the thymus. And B, it's there are bursa-dependent. Bursa present only in birds, but in the human body, present peripheral organs of immune response where B lymphocyte finish maturation. But the surname B or bursa dependent because at the first they were found in research in birds. So T and B lymphocyte, they origin in the bone marrow, and they undergo differentiation as the first in the bone marrow. Every foreign element of the blood origin and finish maturation in the bone marrow and leave the bone marrow only in the 6th class, and except T lymphocyte. In the 3rd class, to jump from the 3rd class of differentiation to the 4th class, to become blast, all foreign element of the blood need special stimulation substances which are named erythropoietins. All foreign element of the blood get these erythropoietins in the bone marrow. Erythropoietins are produced in kidney, and they get to the bone marrow to stimulate hematopoiesis. But T lymphocyte cannot get these stimulation factors. Here they migrate to the thymus to get these factors, because thymus produce thymallin, thymazine, and thymapietin, which necessary for maturation of T lymphocyte. So T lymphocyte finish maturation in the thymus, and it's this part of maturation is named antigen independent maturation, because they never connect with antigen. They don't know what is it antigen. To get the information about antigen, lymphocytes go to peripheral organs of immune response, such as lymph nodes, Peyer's patches, tonsils, and spleen, to finish maturation. In these organs, lymphocyte can get antigen and can get information about structure of antigen, quality of antigen. And macrophages very helpful for maturation, antigen-dependent maturation of lymphocyte. So T lymphocyte, after finish maturation, became T killers, T helpers, T suppressors, T memory cells. There are 4 mature types of T lymphocytes. B lymphocyte also get 2 steps of maturation, antigen independent in the bone marrow an antigen dependent in peripheral organs of the immune response. After finish maturation, B lymphocyte became plasma cells. So B lymphocytes, they circulate in the blood for some days, and after migration to the connective tissue, after finish maturation, they became plasma cells and start produce antibody. And they make and create after that humoral immunity. And T lymphocyte creates cell immunity. So lymphocytes, T and B, they are main immune cells. And macrophages, antigen presentation cells, because they help them to have a meeting with antigen and get information, get knowledge about antigen. And what about next type, a granulocyte, which are monocyte. A monocyte is about 3 till 11% of all total volume count of leukocyte. There are large cells. There are polygonal cells. They originate in the bone marrow, and is from the 6th class. B mature monocytes. They leave bone marrow, circulate in the blood for 3-5 days, and after that they migrate to the connective tissue everywhere in the human body and get new name, macrophages. And in different places where we have the connective tissue, and we have a different type of macrophages. For example, in the skin, macrophages are named Langerhans cells. In the liver, Kupffer cells. In the bone tissue, osteoclast. And in the nervous tissue, there are microglial cells. And the famous Russian scientist Mechnikov researched all of type of monocytes, and which are macrophages, and holds them together in the table, macrophageal table of Mechnikov. And after migration to the connective tissue, monocyte, which became macrophages, take an active part in immunity by its phagocytic function and by antigen presentation function, because macrophages help to be T and B lymphocyte to finish antigen-dependent maturation. Percentage of different type of leukocyte here present, and we call this percentage of different type of leukocyte as a leukocytic formula. So basophils 0 till 1%, eosinophils 1 till 5%, neutrophils 65 till 75%. Don't forget about the classification of the neutrophils, depend on the step of maturation. Lymphocyte 9 till 7%, and monocyte 3 till 8 or 10%. It's a leukocytic formula. And let's go to platelets. Platelets or thrombocytes. Platelets are normally count of platelets 180 till 320, multiply upgrade 9 per 1 liter. A very, very small format element of the blood. It's about 2 till 3 microns, as you see. What about formation? How do they organize? Platelets are pieces of cytoplasm cell membrane of the megakaryocyte. So megakaryocyte, it's a very, very large cell, which is located out of the sinusoidal capillaries of the bone marrow, and they put cytoplasmic processes through the windows, which are pores in the membrane of the sinusoidal capillary inside into the space of capillaries, and the species by the bloodstream are erupted, and the species of cytoplasm of the megakaryocyte get named platelets or thrombocytes. Thrombocyte has platelets divided into 4 zones based on the structure and function. It's a peripheral zone, structural zone, organelle zone, and membrane zone. Depend on the structures which rest here after rupturing from the body of the main cells, after rupturing from the body of the megakaryocyte. And what about function? Main function of platelets is maintenance of homeostasis by formation of thrombi, of clots, blood clots, after the maiden of endothelium of blood vessels. And after the maiden endothelium of blood vessels produce special activation substances which activate thrombocyte, and like feedback they release activation substances for activation of fibrinogen. Fibrinogen became fibrin insoluble, but thrombocyte aggregated on this network of fibrinogen in the place of the maiden of the blood vessels, and here happened formation of the large blood clot, and it stopped bleeding from the maiden place of the blood vessel. Here you can see a new name, new meaning of the topic of the blood. It's a gammogram. Gammogram, it's a count erythrocytes, leukocytes, and platelets per 1 liter of the blood. It's a normal count. We call this count of erythrocyte, leukocyte, and platelets together as a hemogram. So, a little about formation of foreign element of the blood. The process of formation of foreign element of the blood is named hemopoiesis in general. It's happening in the bone marrow. It can be divided, depending on the type of foreign element of the blood, to erythropoiesis. It's a process of formation of erythrocytes. Thrombopoiesis, process of formation of platelets. Granulopoiesis and monopoiesis. And for these foreign element of the blood and processes of formation compose the large group which is named myelopoiesis. Myelopoiesis is a process of formation of erythrocyte, platelets, granulocyte, and monocytes. And process of formation of lymphocytes is named lymphopoiesis. It's an isolated process of formation. The process of hemopoiesis can be divided to 6 classes. And famous Russian scientist Maximov described this process of hemopoiesis, and they explain it like a table from 6 classes. In the first class take place stem cells, which is mother cell, which is the youngest cell, undifferentiated, undetermined. Mother cell of the hemopoiesis. The next class, second class, contains two cells, which are semi-determinated cells. Why semi-determinated? Because if you remember from this, a cell get one of possible ways of differentiation. It's a myelopoiesis or lymphopoiesis. So it's it's half of determinated cells, semi-determinated cells. It's a previous progenitor cell of lymphopoiesis, and this one is a progenitor cell of the myelopoiesis. After that, we have third class, colony building cells. And from the third class, cell can jump to the fourth class under the influence of erythropoietins. Erythropoietins, several activation factors, which mainly produce in kidney, and for T lymphocyte, hemopoietins produced in the thymus. So T lymphocyte migrate to the thymus to continue maturation under the influence of the malin, thymazine, and thymopietin. In fourth class take place cells which are blast. Blast not differentiated, they are determinated, because every blast knows which type of cells finally it became. So they are completely determined, but they are not differentiated cells. Blasts get new functions. They can divide by mitosis to make numerous cells of each type, of it type. After that, they jump to the fifth class. Fifth class, there are differentiated cells. In each fifth class present a lot of kind of one type of cells. They quickly undergo morphological and functional changes. And finally, sixth class. In sixth class, present only mature cells. Only mature cells can leave bone marrow. Only mature cells can be found in the peripheral blood. If in the peripheral blood found young cells, it can tell about oncological disease of the blood. So it's about hemopoiesis. It means process of formation of foreign element of the blood.